So in this video, we're going to spend about half of this talking about the ear, and we're going to spend about the other half talking about disorders of the senses. So let's get into the sense of hearing. First of all, the ear has got two sensory functions, not one, both hearing and balance, which is referred to as equilibrium. And so the reason why the ear is involved in both of these senses is because the receptors for both of these are located in the inner ear, and we'll go through the divisions of the outer, the middle, and the inner ear in a diagram in just a minute. So what these share in common is each of them has hair cells that have what are called stereocilia, and these are long microvilli. So these are extensions of the cell membrane, but these are sensitive to mechanical stimulation, so they're going to be sensitive to any sort of movement of the fluid around them. So this makes them mechanoreceptors. Okay, so just looking at the anatomy of the ear, there are three divisions. The outer ear, which consists of the pinna and of the auditory canal. So the pinna is basically what most people think of as their ears. It's the outside of your ear, and it connects and funnels sound to the auditory canal. In the auditory canal, you've got modified sweat glands that secrete earwax, and the actual purpose of earwax is pretty much to keep insects out. And the reason or the way that earwax keeps insects out is because to them it tastes really bad. So I'm sure that you all knew of at least one kid in elementary school that was the gross kid of the class who just had to taste his earwax. Always a boy, never a girl. And so let me start this conversation out by saying that I was not the weird kid who tasted his earwax. I was the kid who put the weird kid up to tasting his earwax. So anyway, of course, they would taste it. They would say it was horrible, and insects think the same way. So you've probably had insects flying into your eyes, into your nose, into your mouth, especially if you've been on a bicycle. But it's pretty rare to get insects down into your ear, and this is the reason why. So it's kind of protection against insects coming into your ear because imagine um, a decent sized insect getting into your ear, crawling inside your ear, and then ice skating across your eardrum. That's something that would be incredibly painful. Okay, so the middle ear begins at the eardrum, which is also called the tympanic membrane, and ends at a wall with openings called the oval window and the round window. Now, the what connects the eardrum to the round window are three bones that are called the ossicles. And so these are the three ear bones, the malleus, the incus, and the stapes. And so basically, when sound hits the tympanic membrane, it's going to vibrate. And the ossicles actually amplify that vibration and transmit it to the oval window. So along with all of the rest of these, as a part of the middle ear, you have what's called an auditory tube that extends from the middle ear to the nasopharynx. And this permits the air pressure within the middle ear to be equalized. And so when you yawn and you pop your ears, this is when the auditory tube comes open and the air pressure between the middle ear and the outside equalizes. So the inner ear is the only part of the ear that's actually fluid filled, and you've got three areas there. The semicircular canals, which play a role in detecting equilibrium, the vestibule, which also has a role in equilibrium, and the cochlea, which has a role in hearing. So here's what the human ear is going to look like. So if you look at the blue section, this is your outer ear. If you look at the pink section, that's the middle ear. If you look at the yellow section, that's the inner ear. So let's start with the outer ear. Here you've got the earlobe, and then the entire outside of the ear is called the pinna. This is the auditory canal, which leads into the middle ear. So the tympanic membrane or eardrum is the boundary between the outer ear and the middle ear. Taking a look at the middle ear, you've got three ear bones basically. The first one that contacts the tympanic membrane is the malleus, and it's named as such because if you take this bone out, it actually looks like a little miniature hammer, a mallet. And so the Latin name for this is malleus. And what is it that you hit with a hammer if you're a blacksmith? You hit an anvil. And so incus is actually the Latin name for anvil, and so it kind of gets struck by the malleus or gets contacted by the malleus. The final one is called the stapes, and if you kind of turn your head to the side and look at this, the stapes looks like a stirrup, and so stapes is actually the Latin name for stirrup. And so hammer, anvil, and stirrup are actually the common names for these ear bones. Looking at the inner ear, it doesn't show this here in this diagram well at all, but the stapes actually contacts a part of the inner ear called the round window. So this organ is called the cochlea. This is going to contain the hair cells that are responsible for picking up vibrations that are transmitted through the stapes to the inner ear. This is the cochlear nerve, and this is the nerve that all of the hair cells in the cochlea feed into. These are the semicircular canals. Now there's 3 of them, and they're in actually 3 dimensions, X, Y, and Z dimensions. So this allows you to have 3-dimensional equilibrium. This is the vestibular nerve, and you notice that there are 3 nerves coming in. At the base of each of the semicircular canals, you have something that's called an ampulla. And so that's the swelling that's at the base of those semicircular canals. And each ampulla sends impulses into the vestibular nerve. So you have the cochlear nerve and the vestibular nerve, and what these join into, the 2 of them actually join together to form the vestibular cochlear nerve. So the semicircular canals, the cochlea, and the ampullae, these are actually all called the vestibular cochlea. This is the round window, and what the round window does is sort of compensate for the oval window. This gets a little bit complicated, and I'll explain this in a diagram where we've got a little bit more detail. This is the auditory tube. You can see that it's got a connection with the middle ear, and so the auditory tube, again, when it's open, it will equalize pressure between the middle ear and the nasopharynx. So some of the time when you have a cold, then this will actually get closed. So the tissue surrounding the auditory tube will swell and close the auditory tube. And as the auditory tube closes up towards the middle ear, it forces air in towards the middle ear. And this is the reason why you begin to feel really stopped up there. And this is the reason why when you finally get that auditory tube to open back up and you get equalization of pressure, it's a tremendous relief. Okay, so the auditory pathway to the brain is through the auditory canal in the middle ear. So again, our process begins when sound waves enter the auditory canal, and this will vibrate the tympanic membrane or eardrum. Now these vibrations get amplified across the middle ear bones. One thing that can actually happen with age is that the connection between the middle ear bones becomes less flexible, and as a result, you don't get as much of the transmission of the sound, and so you can have hearing loss, especially in the higher frequencies. The stapes touches the oval window. When the oval window vibrates, then it'll set up vibrations in the fluid inside the cochlea, and so so the fluid starts vibrating, literally vibrating within the cochlea. And so here's a more detailed picture. So here's the stapes, and it connects at the oval window. And this is the cochlea, and this is the round window. And so the function of the round window is this. So right here, as the stapes moves back and forth, it's going to move sort of, as the stapes move the oval window, it's going to move sort of back and forth, inward and outward. And as it does this, as the oval window moves in, the round window sort of bulges out. So it acts as sort of a compensator for the pressure that's involved when the oval window gets pushed in. So if you think of squeezing like a long balloon, and as you squeeze one part of the balloon, the other part of the balloon bulges out. It's the job of the round window to actually bulge out and compensate for the change in pressure in the cochlea as this vibration is happening. So these are the semicircular canals, and I want you to notice again that each semicircular canal has what's called an ampulla at the bottom of it. So where does the data go then? Well, as the cochlea gets stimulated, it sends nerve impulses to the auditory cortex. So when the stapes hits that oval window and vibrates it, these pressure waves in the fluid move across what's called the basilar membrane that is lying across hair cells, and we'll see that in more detail in another slide in just a minute. Okay, this basilar membrane moves up and down, and stereocilia that are in the tectorial membrane bend. And again, we'll see this in more detail in just a minute. Nerve impulses begin in the cochlear nerve and travel to the brain, and once they reach the auditory cortex in the temporal lobe, this is when they're interpreted as a sound. Now, one thing that I can tell you is that I don't believe that the cochlear nerve actually that the vestibulocochlear nerve actually goes through the thalamus. I think it goes directly to the temporal lobe. Okay, so this is the basilar membrane right here in blue. Okay, so this is the basilar membrane. That's what actually has hair cells in it. So basically the way that sound gets transmitted here is the tectorial membrane is actually moved back and forth by the pressure waves coming in. What this is going to do is it's going to bend these stereocilia right here. So these stereocilia are going to move back and forth. And what that movement back and forth is going to do is actually set off the hair cells. So it'll set off an action potential as these get moved. The hair cells will actually move. Oh, I'm sorry, the hair cells will start an action potential, and this will send nerve impulses. These will send nerve impulses down their axons as these get moved to the cochlear nerve. And this is how sound basically gets to the brain. Okay, so the next thing we're going to talk about is equilibrium. And you've got three semicircular canals, and these are arranged in such a fashion that each one is in each dimension, x, y, and z. So each semicircular canal, as I said, has an enlarged base called an ampulla, and the ampulla actually contains the hair cells with stereocilia. So as fluid within these canals flows and bends a cupula, the stereocilia within it are bent, and this changes the pattern of impulses that are carried in the vestibular nerve to the cerebellum and to the cerebrum. And the brain uses this information to make possible corrections. So here's the cochlea, here are the semicircular canals, and these are the ampullae, and here is a receptor in an ampulla. Here's the vestibular nerve, and you can see that there are nerve fibers that are running from each ampulla into the vestibular nerve. Okay, now this is sort of a blow up of what's happening in the ampulla. So you've got liquid that flows across it, and this liquid flow is going to change based on the change of flow in the semicircular canals. And so why have the canals at all, these loops sort of reaching out? It amplifies the sensation of movement by making it easier to move the liquid. So when that liquid flows, the cupula is going to bend, and that's going to bend the stereocilia like here and here and here. Okay, so these stereocilia get bent back and forth. And what that's going to do is that's actually going to stimulate the hair cells, which are then going to stimulate this, as with hearing, you're going to stimulate this basilar membrane that's right here, and you're going to see transmission of action potentials, in this case to the vestibular nerve. And so the supporting cells here are actually not cells of perception. They're just there to form a scaffolding for the hair cells to rest on. And there's the vestibular nerve. So you can see as the liquid flows that the stereocilia themselves are also being bent. And as they're bent, then they're going to transmit this sort of change of direction. And you can see right here in the diagram that the guy is shaking his head back and forth. Okay, so if you spin around and around, the reason why you get dizzy afterwards, where you can't really walk straight after spinning around, is because the liquid is continuing to flow. So when you stop spinning, the liquid doesn't stop flowing immediately. It continues to flow, and that's what gives you the sensation of dizziness. Okay, so we're going to spend the rest of this PowerPoint talking about disorders that affect the senses, and we're going to start out with disorders of taste and smell. And so your sense of smell can begin to decline after age 60, but there are some people that are born without a sense of smell, and this is called anosmia. There are other factors that can contribute besides age to a decrease in the ability to taste or smell. One of them can be upper respiratory infections, so infections that affect the pharynx, the nasopharynx, the oropharynx. Allergies can contribute to this, and so can exposure to drugs or chemicals, including tobacco smoke. Now, one of the things that can happen because the epithelium that detects taste and smell is because it's epithelium, sometimes you can get a little bit of regeneration of this. Sometimes if you lose your sense of smell due to a disease or some sort of infection, sometimes it can come back. One of the things that's actually being noted with the coronavirus is that people lose their sense of smell temporarily, and that can be an early cue to the fact that they've actually got the virus. And brain trauma can contribute to a decrease in the ability to taste or smell, in that you may have perfectly functioning organs of smell, but the brain may not have the capacity to receive them because it's been damaged. Okay, disorders with the eye, color blindness and problems with the focus of your vision are two common abnormalities of the eye. More serious disorders can result in blindness. So let's talk about color blindness first. The first thing to tell you is that complete color blindness is actually rare. Um, in most instances of color blindness, remember that there were three types of cones, red, blue, and green. In most instances of color blindness, you have a particular cone that it's either lacking or it's deficient in number. Of color blindness, red-green color blindness is your most common type, and this is actually a genetic trait. It is on the X chromosome, and it is recessive. It's an X-linked recessive trait. This is going to affect between 5 to 8% of males and between 0 to and about 0.5% of females. And the reason why it affects more males than females is because males only have one X chromosome. And if you only have one X chromosome, you don't have a second chromosome to compensate for it. So females have got two X chromosomes. If they have one X chromosome that carries color blindness and another X chromosome that is a normal X chromosome, the normal X chromosome can compensate for the abnormal one. So the reason why there's only 0.5% of females that have red-green color blindness is because they have to have both of their X chromosomes to be the mutated ones in order to be red-green color. Okay, nearsightedness is a problem with visual focus, and this is when you can see close objects better than distant ones. And the reason that this happens is that the eye itself, the eyeball, is slightly longer than what it should be. So when the lens tries to focus images on the retina, it actually focuses them at a point in front of the retina. Now, what this is going to do, this can be corrected by concave lenses, and what concave lenses will do is to spread the light rays out so that when the lens focuses them, that focal point reaches further back to the back of the eyeball. Farsightedness is just the opposite, and this is when you can see distant objects better than close ones, and usually in this case the eyeball is shorter than usual, and that means that the image gets brought to a point focus behind the lens. And so this is going to be corrected by convex lenses, which increases the bending of the light rays, and the focus comes forward to hit the retina. So if you look at this with a long eyeball, you see where the dotted line is on the left here. So at this dotted line right here, this is where the normal length of an eyeball is, and this is where a normal shaped lens here, the normal shaped lens would normally focus right here. And so what this concave lens does is it spreads out. It actually spreads these rays of light out. So when they hit the eyeball, they're spread further out, and this means that they converge at the proper distance back here. So in astigmatism is when the cornea or lens is uneven, and this produces a fuzzy image. And in this case, the light rays are not evenly focused on the retina, and you can wear a lens that's unevenly ground. So the lens has sort of matching defects to the cornea or the lens, and this compensates for the uneven cornea. So right here, basically, you can see here that the light rays are kind of getting tangled up, and this is because of an irregularity on the cornea. And so here, if you have this sort of, if you have this unevenly shaped lens here, then what that's going to do is kind of straighten out. It's going to compensate for and straighten out the light rays so that when they hit the back of the eyeball, when they hit the retina here, they're hitting in a normal formation. Okay, so the next thing we're going to talk about is retinal disorders, and this is something that I mentioned before, diabetic retinopathy, in which capillaries to the retina can become damaged. And when this happens, you can get blocked vessels and hemorrhages. And what's happening there is actually that you get rods and cones that are not receiving, they're not receiving their normal blood supply. Another thing that can happen here is that you can get some edema or swelling. In addition to the lack of oxygen supply, another thing that can happen is edema or swelling due to either one of these. And this is actually something that I had to deal with in December and January. I had, because of some higher blood glucose levels back in the fall, I actually had a small artery in my left retina actually burst, and that caused some swelling. And I had to take some medicated eye drops for quite some time in order to try to get that cleared up. And at my last eye appointment, the swelling was still present, but not as distinct the swelling. So macular degeneration can happen when you get the destruction of cones because thickened choroid vessels no longer function. And so basically, if the vessels in the choroid are not functioning, then they're not going to send capillaries to the retina, and that's going to lead to the death of cones. Okay, in retinal detachment, following some sort of physical trauma, the retina gets torn or separated from the choroid. And one of the reasons why I really wish you could have done an eyeball dissection is because you'd be able to see how easily that the retina comes away from the layers that are below it. And so this is something that can happen to outside trauma. So you'll hear of boxers and football players and professional wrestlers, people who are in high impact sports or professions, they can actually get their retinas detached. Okay, glaucoma happens when fluid builds up in the eye because the drainage system fails. One of my favorite names in biology, because hardly anyone hears of it, is the canal of Schlemm. And the canal of Schlemm actually drains the eyeball. And if the canal of Schlemm gets blocked, as it can in glaucoma, then fluid can build up in the eye. And this fluid buildup will put pressure on the retina, and it can destroy nerve fibers that are associated with peripheral vision. So someone with glaucoma will lose the peripheral vision first, and then it'll kind of come inwards. Cataracts are cloudy spots on the lens that can eventually cover the entire lens. And your risk factors for cataract can be a lot of exposure to UV light. So basically, if somebody's out in the sun too much, skiers can have cataracts, fishermen can have cataracts. Diabetes is another one. I've got lens replacement in my eyes because diabetes caused cataracts with me. Heavy alcohol consumption and smoking. And so I just got to be honest, for me, the worst part of having these cataracts was that I still had extremely good vision for my age. I didn't need reading glasses or anything. And then the cataracts happened, and they clouded the lens up so badly that I couldn't tell the difference between one student and another at the back of the classroom. And so in the lab room 116, I would have been able to tell the difference between students in there. But in any of the other classes where you've got like a normal sort of square-shaped classroom, I would not have been able to tell the difference between students at the back of the class. I also had a ton of trouble reading assignments and so forth, and just had to have one lens replaced and in October, and then the other lens replaced in December, and this was back in 2017. Okay, so this is what a cataract can actually look like. Okay, hearing loss can develop gradually, or it can be sudden, and it's got a lot of different causes to it. 1 in 3 people over the age of 60 have got hearing loss, and part of this can be due to middle ear infections, and they're subject to that because The reason why the middle ear is subject to infections is because it's got a connection, if you'll recall, with the pharynx, and the pharynx is connected to the outside environment. So this means that the middle ear can be subject to infections, and if the infections stay there, then you can get hearing impairment if they're not properly treated. And so the first sign of hearing loss is having a lessened ability to distinguish between conversation and background noise. Hearing problems can arise as early as age 20, but as I mentioned before, the mobility or the ability of the ossicles to vibrate can decrease with age. Okay, sudden deafness can occur, and it usually occurs in one ear, and the causes can include infections, trauma, side effects of some drugs. I had something called viral labyrinthitis, which actually was a viral infection of the vestibulocochlear organ. And when you have that particular disease, it came along as a companion to the flu. So sometimes when you get the flu, it'll bring a buddy along. And in this case, the buddy attacked my ear, my inner ear. And you can have hearing loss, permanent hearing loss, or you can have severe vertigo, or you can have both. And I had a little bit of hearing loss in that ear, just a little bit. Higher frequencies or something I have a little bit more trouble with in that ear. But the main thing that brought on was severe vertigo, and the vertigo lasted in one degree or another for about 8 weeks. Okay, it can resolve itself. Unfortunately, in my case, the vertigo did resolve itself. The deafness did not. You can have deafness at birth, and this is usually due to a genetic disorder. But sometimes, if a mother gets infected with German measles or the mumps during pregnancy, this can cause deafness at birth. Okay, so this is what I mentioned, disorder of equilibrium, and vertigo is the main one. And this is definitely a feeling that the person or the environment is moving when there's no motion. And so I could be lying on my back in a bed, and it would feel like the bed was coming up and over me. I would feel like I was tilting forward, and there never was a time period in which it felt like the world stopped moving. So I couldn't walk straight, I couldn't drive, and tracking motions with the eyes would make the vertigo a lot worse. So anytime that I tried to read, I would just about pass out. At the time, there weren't any flat screen TVs. They were all CRT monitors. And the way that an old style monitor and television worked, the tube TVs, is there is one single point of light that moves back and forth across the screen, but the point moves so fast that you perceive it as a single image. But subconsciously, I would perceive that. So anytime that I looked at a computer monitor or TV, I would just about pass out. So couldn't read, couldn't look at a screen, and so I had this for 2 weeks in the middle of a semester. It was at its worst. I missed 2 weeks of classes when I was in a PhD program, and it took me a lot of work to get caught up with that. And so, but what I had to learn to do was to ignore the input from my inner ear. So I had to learn to ignore the vertigo in order to be able to walk normally and drive normally. But for a few weeks, when I was walking down the hallway, I would walk right next to the wall, and I would have a hand on the wall to kind of steady myself so that I wouldn't weave and stagger. So yeah, this is stuff that can be pretty bad. And if you're prone to motion sickness, if you have vertigo, you're going to be throwing up all the time. Fortunately, I'm not real prone to that, so it only made me nauseated a few times. The other thing about this is that I learned at that point to, like I said, ignore the input from my inner ear, and it means I am immune to seasickness, because if the motion's disturbing, I can tune it out. Again, this can be caused by problems in the inner ear, and that was the problem, or by problems in the brain. And the way that vertigo usually resolves is that your brain learns to reinterpret the altered data coming in from the inner ear. So with some damage to the vestibular nerve, what was happening was the signals that were coming in along that nerve were kind of altered. And so my brain had to relearn how to interpret signals coming in, and that eventually canceled it out. There's something that can be called benign positional vertigo that's due to particles in the semicircular canals, and so that can alter the way that the fluid flows, and that can alter your